Morning, everybody. It's December 3rd, and we're very fortunate today to have Dr. Ed Lean from the Allen Neuroscience Institute give us a talk on single-cell molecular approaches to studying and genetically targeting human brain cell types. And Dr. Lean has actually been a great partner with us here at Swedish. He's a senior investigator at the Allen Institute for Brain Science. He's got numerous different affiliations. He originally did his undergrad in biochemistry at Purdue and PhD in neurobiology from UC Berkeley. Actually performed a postdoc fellowship at the Salk Institute. And he joined Allen back in 2004. He's done some really seminal work, and basically for me as a neurosurgeon is really understanding what cell types are in your brain and how they all communicate. And he's done it on a large scale. He's also looked at gene expression, and he's doing some really amazing work at the Allen Institute. And with the research there, has worked with our group on several projects. And I could go on and on and on. And we really are fortunate to have Ed as a colleague, but also just as in the scientific community. I think for most of the neurosurgeons, I can say this is really understanding anatomy is so important to what we do. So Paul Allen, I think, was such a visionary person, but to actually fund this whole project and to really try to understand what goes on in the human brain is fascinating to me. So without further ado, Ed, thanks so much for joining us.
Thank you very much for the kind introduction and for the opportunity to speak. Let me try to share my screen here. Okay, does that look okay?
Looks great.
Okay, great. Again, thanks for the opportunity. I'd like to really share with you some of the progress that we've made in trying to characterize the defined cellular complexity of the brain, which has actually led very directly to new tools that may actually have clinical significance as well. So let me begin by saying that our grand sort of goal at the Allen Institute was to take a very reductionist approach to try to break the nervous system down into its components. And those components are cells, which we can try to abstract into a classification and abstraction down to cell types that have consistent and coherent properties that we can characterize and classify. And then think of the nervous system as a network built out of those types of cells. This is a fairly challenging thing to do, given the complexity of the brain, and also particularly challenging to do in the human brain. On the mouse, of course, I think all of us are very familiar with the many things that make the mouse an appealing model system and have made it the dominant biomedical model system. You have very good access to genetically targeted cell types that give you cell type specificity. So the level of granularity that we understand the mouse, or beginning to understand the mouse at least, is extremely high. And understanding the human has really lagged behind this for a variety of reasons. The limited accessibility of the tissues, the inability to bring to bear genetic tools for being able to both target and manipulate certain kinds of cells. And so there's really been a big sort of chasm between our degree of understanding of the human versus model organisms. So what we've been trying to do is to bring to bear modern technologies to both understand the mouse brain at very high resolution, but also then to translate these technologies to understand the human, so we can really evaluate what the state of the human brain is, and also how well this is modeled by other model organisms such as the mouse or monkey. This has led to really trying to push a whole series of technological approaches. Some of these are relatively mundane, like the proper optimization of tissue preparation to be able to use various types of technologies on human brain tissues. This is actually pretty variable in the field, but there are ways of being able to prepare these tissues so that they work with all of the latest technologies. And the really transformative technologies that I'll spend a lot of the time on today are the single cell omics methods. These have really transformed the field in short order by the ability to isolate individual cells and profile the genetic content of those cells. This has now been translated to work on human brain tissues or other model organisms that are more difficult to work with by looking in the nucleus rather than the cell. And that allows the use of frozen post-mortem tissues for single cell transcriptomics and epigenomics, as I'll explain, as well as the new versions of spatial transcriptomics. We've also been pushing this to beyond the molecular characterization to the functional characterization of different kinds of cells. This has involved our colleagues at Swedish Hospital and in the UW system, as I'll describe later, to try to be able to use tissues resected in the course of neurosurgeries to study the functional and anatomical properties of cells, and then to connect that to the molecular. So I'll describe some of that. And then finally, an unexpectedly successful approach has been to take these molecular tools and begin to develop molecular genetic tools that allow the targeting of particular types of cells with viruses and cell type specific enhancers to be able to do very targeted manipulations that work across species, including human. So this whole approach has been really quite dramatically accelerated by the advent of a whole series of big consortium efforts that all intend to create atlases or censuses of cell types in different organ systems. We are a big part of the NIH Brain Initiative Cell Census Network, or the BICCN, which is a very big effort to try to create a map of cell types across the entire brain in mouse, monkey, and human. My colleague Honghui Zheng is actually now the leader of the Allen Institute for Brain Science, runs a big project to try to create a brain-wide atlas in mouse. I run one now to try to do the same thing in human. And our colleague Mike Harlitz at the Allen Institute also runs what's called the Brain Cell Data Center, which is the data hub for the entire consortium to make these data publicly accessible for the community. There are also efforts in many other organs, and in particular an effort to try to coordinate across all the organs called the Human Cell Atlas that we also participate in. The basis for basically all of these census efforts is that we can use new technologies to be able to classify and define the cell types that make up the brain. Traditionally, cell type classification in the nervous system has been based on anatomy and function. And the new technologies that I'll be describing today for the molecular side really kind of turn this around to have a genetic paradigm for defining cell types. And the basis of this is that the genes that are used differentially by different types of cells are what give the cells their different properties. And if we can measure all the genes, then we can classify and we can cluster quantitatively based on those gene expression profiles. So the technologies have really enabled this now of single cell transcriptomics, where it's possible to measure somewhere between 6,000 to 10,000 genes per cell across many thousands of cells. And now we're starting to talk about millions of cells that are available for these types of classifications. And this is really kind of changing the field pretty quickly to trying to move sort of in the direction of genetics, where first there was a lot of discovery, and then it started to move into a more defined classification and nomenclature system. That's sort of the transformation that's happening now, that the discovery phase is coming to an end in certain parts of the brain. And now we're starting to move to rigorous classification schemes and nomenclature schemes to try to get the community to talk on the same page. So the basis of this approach is as I've mentioned already, is to cluster on the basis of genetic expression profiles. And so the basic idea is that you can take regions of the brain and dissociate those into individual cells. And now this works with individual nuclei of cells. And then create a library based on the RNA in the cells, and then sequence that library. And there are multiple methods for doing this. This actually started with this idea of measure the very detailed full-length sequences in cells, and this has sort of now been taken over by a shallower sequencing that allows you to measure many more cells. The 10x Chromium platform in particular has really changed this field by allowing much greater throughput for much cheaper costs, so that one can generate a classification based on tens of thousands to even hundreds of thousands of cells. So illustrated on the right is a dimension reduction plot, taking this say 20,000 gene space and reducing it to 2 dimensions. So you can see that these different clusters based on the gene expression profiles represent putative cell types that then we try to characterize and understand what the properties are. So the utility of this is that we now really kind of have bounded the complexity of the system so that we can begin to ask, what are the properties of all these cells? And part of the talk today will be describing some of those efforts to begin to move from this sort of abstract molecular classification to understanding the properties of these cells, the physiology of the cells, the connectivity or the function, and the anatomy. And as I've hinted at already, there's really a very direct path from a molecularly based classification to tools to manipulate based on that molecular classification. So this whole effort has really been put on steroids by this NIH Brain Initiative Cell Census Network. We have brought together a whole series of top labs to bring their respective technologies to be able to contribute to this characterization and build a robust classification. And it's really a very interesting initial phase of this project where we were deciding where we should put the initial emphasis. And we decided to take a page out of out of genomics, where the ENCODE project picked 1% of the genome to characterize with a whole variety of different techniques. The BICCN decided to pick 1% of the brain, one brain region, to do this initially, and that region is primary motor cortex. And we have groups that focused on the mouse, on the marmoset monkey, and human, using these single cell methods that I've described for the omics, spatial methods to look at the distribution of cell types, functional methods to look at the physiology and anatomy, and a variety of tracing types of methods to look at the connectivity of these cells, to really put all of this together. And the outcome of that is a really robust classification of types in primary motor cortex that serves as the template for how we can approach the rest of the brain. And I'll spend a couple of minutes on this because this is really the basis now for the rest of the talk. This classification can be represented as a hierarchy, and the upper nodes in the hierarchy differentiate the main cell types. We can differentiate the neurons from the astrocytes and oligodendrocytes and various other non-neuronal types. Within the neurons, the next level in the hierarchy are the GABAergic cells versus the glutamatergic cells. The next level would be, for example, the subdivisions, we call these subclasses of the interneurons. So these are the somatostatin or parvalbumin positive or VIP or LAMP5 groupings. And actually, these groupings really correspond quite well with the developmental origins of these cells, the interneurons that migrate in from the ganglionic eminences during development, whereas the excitatory neurons are developed in the cortical plate. Along with the sort of classification, we get the relative proportions of the types of cells. And what I think you can see on the bottom of this plot here, this histogram represents the relative frequency. The most of these cell types are actually rare in the cortex. There are a few that are more prevalent. But this is sort of the nature of the system, is many discriminable, highly granular types of cells at low proportion. We also now have a lot of understanding about the projection targets of many of these types of cells. So we know whether these are cortical, whether they project to deep structures, and where they are located in the cortex. So this really now forms the foundation for aggregating information about cell types that becomes an increasingly rich knowledge base to then understand the system. Another important element of this is that it is really quite a statistically robust classification that is consistent across a variety of different technologies. So there are multiple different ways of measuring the transcriptome. There are different technology platforms. These are all quite consistent with one another. It's possible to align cell types defined by the genes across technology platforms. And it's also possible to map between the transcriptome and the state of the chromatin, the epigenome. And this really begins to align the epigenetic landscape with the active gene transcription in different types of cells. And kind of remarkably, these are, well, philosophically, these should be well aligned, but that doesn't always turn into reality, but they actually align very well so that we have a good map now of the gene expression along with elements of the epigenome that would be consistent with those genes being turned on. So for example, hypomethylation of active regions of chromatin, and those regions are open chromatin rather than bound up in the nucleus. So very, very robust classification. Now, a particularly useful feature of this molecular sort of approach is that it's a very rich information set. And that allows us to use that to align data sets from disparate data. I've just described to you that you can align across different methods. You can take this in different directions now too. One of which that we've really helped to pioneer is to align them across species. And we call this homology mapping, where we can take these molecular definitions, these molecular clusters, and then align them across the species to have an aligned representation. And this allows us to map together the data from a mouse and a human so that you can see just which cell types are very homologous, which may be more divergent, and what their properties are. And I'll just give a hint that also this same approach we're now applying to disease, where we can align disease data sets to a neurotypical reference and start to look for deviation from the norm as the disease state. So through this Brain Initiative project that I was describing, we've now been able to take this to 3 species between mouse, marmoset, and human. And in each of the species, we get a very similar answer if we look within species. So that we get a classification of types that is actually remarkably similar in its diversity across mammals. And then we can align these between the different species. And in the middle plot here is a confusion matrix between human and either mouse or marmoset. And the point I want to just show is that this is really along the diagonal, which means that most cell types really align well across the species. So really, to me, a surprisingly high degree of homology between the species. And allows us to create a consensus classification or taxonomy. Now that taxonomy is a bit lower resolution when you try to align across these 3 species, so it's not quite as complex. There's a level of granularity that is in fact species specific, but at pretty far down this hierarchy, we're able to align cell types across species. And this becomes particularly salient for understanding the human, because there are many properties of cells that it's not easy to measure in the human, but we can infer them based on homology. So for example, we have no ability to map long-range projections in human brain at the moment, but we can infer that this particular cluster, which is mapped across species, and we know is the long-range projecting GABAergic neurons in the mouse, is very likely to be the same type of cell in the human. And the same goes for all of the other different types of cells. So this now allows us to really compare across species, and that's where things get a little bit more complex. So I've described this so far as a high degree of homology, where cell types can be aligned across species quite well. But homology is not identity, and there are many examples now of divergent gene expression in homologous cell types. And of course, this is not surprising from an evolutionary point of view. We share limb structure across species, and yet very different functions of our limbs. But this is what we're seeing at the level of cell types and the level of genes now as well. So very frequently, a particular gene will have a different cellular usage. So for example, this gene prodynorphin is in excitatory neurons in human and inhibitory neurons in mouse. When we compare the mouse and the human cortex, among the most divergent gene families with respect to their cellular distributions are actually the serotonin receptors, which have many differences represented here in colors that are either higher or lower in mouse versus human between species. So we now have this information to actually compare apples to apples, and it turns out many genes are conserved, but also many have divergent patterns. And now we can ask this question across the 3 species, and I'm showing here an example of the inhibitory interneurons. We can align these across the species, and then ask what is it that's specific genetically about particular subclasses of these cells in each species. And so, for example, if I could direct your attention to the upper right, this LAMP5 subclass of interneuron, there are about 200 genes or so that are enriched in that class versus all the other interneurons in human. Roughly the same numbers in each of the other species. But a real surprise is that when we ask what the overlap is actually quite small. So there's a core conserved element of this genetic profile, but then there's a lot of species specialization. And I'm not really showing it here, but as you might expect, the more similar, the closer you are evolutionarily, the more similar those profiles are. But it was really kind of quite a surprise that the degree of divergence, that there are more species-specific cell type selective genes than there are conserved genes. Now, we've actually tried to turn this into a bit of a feature. So it's very difficult often to interpret this kind of data. You have so much information. If you want to ask what are the transcription factors that are likely to define the properties of a particular type of cell, the answer is, there are 50 different transcription factors, and it's hard to know how to interpret that. So this evolutionary comparative approach actually helps to interpret the data because it hones in on a set of highly conserved genes that are likely to be responsible for the highly conserved characteristics of the cells, whereas we also have the human profile that actually tells us what the human state is. And to give you an example of that, the famous chandelier cells are a very distinctive and highly specialized type of cell. They are a fast spiking GABAergic interneuron in the cortex, but they have a very distinctive axonal phenotype that they form these cartridges, as they're called, across the cortex. Their other name are axonic cells. They actually target the spike firing or axon initial segment of the excitatory cells. It can have a very potent inhibitory effect. And that anatomical feature is extremely well conserved. So if we take the same approach I was just describing and look for the overlap among the 3 species, it's actually a very small number of genes that is consistent across the species, about 25 genes. And among them are several transcription factors that are likely to be responsible for these core phenotypes, as well as a gene associated with the formation of specific connectivity that forms a pretty good candidate now for why these cells target that particular region on a pyramidal neuron. So we've now really begun to start to use this comparative axis as a way to better interpret and understand what's well conserved across species. Okay, I'd like to shift gears now to beyond the sort of molecular characterization to now discuss some approaches that we've had to really try to understand the properties of human cells. This has really been a fantastic collaboration between the Allen Institute and many of our neurosurgical collaborators within the Seattle area, both at Swedish and at UW. And the idea really was to try to adopt methods of slice physiology to characterize the properties of cells in living tissue slices to work on human tissues. And working with our colleagues, we've been able to get access to to neurosurgically resected tissues from either epilepsy surgeries or tumor removal surgeries. And the sort of typical piece is maybe a half cubic centimeter to cubic centimeter piece of tissue. These are often quite healthy pieces of tissue that can be sectioned on a vibratome as one would for typical slice preparation. And these tissues are very healthy. They allow us to apply a variety of different methods, both acutely. We can record from these for up to several days, and also surviving in culture. And in culture, we can keep these types of tissues alive for several weeks. And that actually allows us to apply molecular genetic tools that I'll describe later in the talk. But the challenge was, how can we try to generate these measurements in human, but also how can we connect those measurements to this molecular framework that I've been describing? And so to do that, we had to come up with a variant on the classic technique of patch clamp physiology, which is now called Patch-seq. We weren't the first to develop this, but we really have kind of industrialized this approach. And the basis of Patch-seq is we would like to get 3 different pieces of information from each cell. We'd like to get the intrinsic membrane properties, the physiology of the cell. We would like to get the anatomy of the cell, and then we would like to pin a molecular phenotype on the cell. And so basically you have a full glass pipette, you form a tight seal on a cell. This allows you then to inject current and record the responses of the cell. The pipette is filled with the dye biocytin, which allows us to fill the cell and later capture the full anatomy of the cell. And at the end, we extract the contents of the cell, in particular the nucleus of the cell. And that allows us to sequence the contents of the cell and to map back onto this reference classification. So to give you one example of this, this was a remarkably good experiment here. In one section of temporal cortex, one of our top physiologists was able to record, fill, and get a molecular profile on 10 neurons here. It gives you a little bit of idea of the spectrum of different shapes of these cells, different firing properties of these cells, and then the molecular properties of these cells as well. So we've been working to apply this very systematically to try to build a reference base of what all the different cell types of the cortex are, which becomes a tractable problem if it becomes characterizing the properties of this molecular taxonomy, since it's a defined set of types. Now I'd like to share with you results from just one part of that study, the first part, which is in the upper layers of the cortex, the excitatory neurons in the upper layers of the cortex. So I think many of you are probably well aware that the cortex has expanded quite significantly between mouse and human. And the cellular diversity is quite apparent to the eye in human compared to mouse. In mouse, these upper layers are supragranular layers are referred to as layer 2-3. And within layer 2-3, the cells are actually pretty uniform histologically. And we also see from the molecular characterization, the transcriptomics, that they also are pretty similar transcriptomically. There's one sort of big cluster that you can differentiate different parts of that cluster, but they're quite similar. In contrast, the human has become much more complex. With the expansion in the depth of the cortex, one can differentiate a clear layer 2 from a layer 3. And the cells get increasingly large as you go deeper in the cortex. Some of the cells in the very deep part of the cortex are these extremely large pyramidal neurons, among the largest in the cortex. And concordantly, the transcriptome also predicted increased diversity compared to the mouse. You can, just to give you sort of this idea, this dimension reduction you can see is significantly more complicated than the mouse, with different clusters representing putative different cell types, and a real expansion or heterogeneity within some of these clusters as well. And so we basically have been trying to characterize each of these types of cells using this Patch-seq technique. And the bottom line from this is that each of these transcriptomically-defined cell types has differentiable properties within the cortex. They're differentiated by their size and their anatomical location in the cortex, the anatomical features, as well as the physiological properties of the cells. Even this sort of elongated sock that I showed you, one of the types has a lot of internal variation. This is also replicated with the various other properties of the cells. So it really seems that this molecular characterization, the features based on the genes, is reflected in the other properties of cells. And so this becomes a really great sort of foundational basis for understanding cell types. Now, the other thing I wanted to point out is that this classification here actually came from recordings from about 100 individual patients. And so that's really quite a remarkable stereotypy, given that these were all derived from neurosurgeries with many uncontrolled axes of variation. But this sort of human blueprint is really well preserved and robust. We've now started to really also try to use this as the basis for understanding disease phenotypes. Just to give you one example of this, a number of years ago, actually several decades ago, Patrick Hoff described a selective vulnerability in Alzheimer's disease based on the tools available at the time. He had an antibody called SMI32, which labels a particular form of neurofilament protein. And he showed that this labels selective types of neurons in the cortex, and that these neurons are selectively lost over the course of Alzheimer's disease. So we can begin to connect this kind of finding with this molecular classification. And so we combined in situ hybridization for these different transcriptomic types with this SMI32 antibody, and found that indeed only a subset of these molecular types label for the SMI32 antibody. In fact, one of those types is one that is not found in the mouse supragranular cortex. So hopefully this sort of foundational reference will serve this purpose in many different disease states, where we can begin to understand what are the vulnerable populations and link it to the genes that may be responsible for that vulnerability. Okay, I'm going to shift gears a little bit now. This is sort of describing the transcriptome. The other approach that we've been taking lately has been to probe the epigenome more specifically. This method, like the transcriptome, has really dramatically accelerated in the last couple of years. And in particular, one method has been very useful for this. And this is a method called Patch-seq. What Patch-seq does is to look for regions of open chromatin. Open regions of chromatin are active, whereas or could be active at least, whereas many other parts of the chromosomes are tightly bound up in the nucleosome and not active for transcription. Within those open regions are both genes that are being actively used and putative regulatory elements as well. So this method has now been scaled down to single cell level, and that allows it to also go to very, very high throughput now as well. And so what one gets from that is a whole series of regions of the chromatin that are active in different cells, and one can cluster on the basis of that, just like I described for the transcriptome. And then because regions of open chromatin contain active genes, it's possible to align this epigenome to the transcriptome, so we know what these clusters represent, that they might represent the parvalbumin positive GABAergic cells, for example. And the reason for this preamble is to say that we can now have genetically defined cell types where we can find cell type specific regions of open chromatin that represent putative regulatory elements for selective gene expression in that particular type of cell. And we've been now pursuing this as a potential way to develop tools to target these cells. So the basis for this really is to use a virus with this enhancer hooked up with a reporter gene that allows us to infect cells in the brain and then see whether that enhancer is capable of driving functional gene expression and driving it in specific cell populations. This has really been accelerated by the development of new viral capsids that are extremely effective at infecting cells across the brain. In particular, one that we use a lot is called PHP-EB. This was developed by Viviana Gradinero at Caltech. And this was designed to be a blood-brain barrier penetrating capsid, so that it's possible to do a venous injection, and it goes throughout the body and throughout the brain, and is able to cross the blood-brain barrier and infect cells very widely. And to illustrate this, on the upper left here is a pan-neuronal enhancer that is labeling a large proportion of neurons across the entire brain. This can now be restricted with more selective enhancers. So in the upper right here is a DLX enhancer that derives only forebrain inhibitory or GABAergic neuron expression, and nothing at all in the deeper parts of the brain. On the bottom are examples of selective expression in astrocytes and in oligodendrocytes. So you can see that this is sort of generalizable, that there are enhancers that can target very different kinds of cells. And before I move forward, I want to just say, you know, as we started to get to this point, it started to become clear that these could be really useful tools, both for basic research, but also for clinical applications. I think many of you are probably aware that the first gene therapies for CNS disorders are now on the market for treating retinal dystrophy and spinal muscular atrophy. At the moment, these are typically gene replacement strategies without any cellular specificity. But you can imagine that if we can begin to target different cell types, that we could do much more targeted therapies using these types of tools to target just astrocytes or just oligodendrocytes, for example. So, or perhaps we could start to push this to even finer cell types if we could understand what cell types are associated with diseases. And so we've been pushing this down from the sort of class level that I just described to now starting to move to cell subclass and type levels of resolution. So here on this slide is an example of pushing this for one of the main types of inhibitory neurons in the cortex, the fast spiking parvalbumin positive interneurons. These are the basket cells and the chandelier cells I was describing earlier. And really quite remarkably, we have been able to identify enhancers that give almost entirely specific expression only in this parvalbumin positive populations, which we can validate both by combining with in situ hybridization. We can use these tools of transcriptomics to isolate the fluorescently labeled cells and then profile them and map back to the transcriptome. So in the middle here, you're seeing the proportions mapping to different transcriptomic types. Interestingly, these enhancers can deliver significantly more selective expression than the genes themselves. So in the mouse field, for example, typically one would make a transgenic animal targeting the gene parvalbumin as a way to hit those cell types. But parvalbumin is expressed very widely across the brain. These enhancers can sometimes give, just the cortical parvalbumin positive cells and not the rest, for example. So there's really a high degree of specificity that can be obtained with this type of approach. And furthermore, these can potentially be used across species. And so we've now been trying to push these tools that are first screened in the mouse and start to use them across species, in particular moving into primates. We've started to use macaque as a primate model organism. And I should say we're also using those human slices as well, but I'm not going to show data on that today. And so this slide is representing the same three enhancers I just showed in mouse, now used in the monkey cortex. And all three of them have conserved cell type specific expression in the monkey that's quite similar to what we saw in the mouse. And the two things that I really wanted to point out here is that the specificity can be very, very high, conserved across species. The top one had 98% specificity for this particular kind of cell. And also completeness of labeling. These are very infectious viruses, and so we can get up to close to 90% of the completeness of cells as well. So this has a lot of implications, I think, for potential uses. If this works in mouse and monkey, we're no longer limited to those species. These probably work across many different species and could expand the range of model organisms to use, but also could have clinical applications. And this is not a sort of fluke that, that we can get this to work across species. Here are a whole series of examples of an astrocyte selective enhancer that gives similar patterning from mouse to monkey, oligodendrocyte, different types of interneurons, different types of excitatory neurons, where this enhancer has very conserved patterning across species, which suggests that the regulatory machinery, the transcription factors and their binding sites are quite conserved at this cell type level. Now, this is not always the case. There are many examples of enhancers, just like the genes, that have that give divergent patterning, that do not give conserved patterning across the species. So we really have to go to the trouble of directly testing these. But there are many examples where we actually can get the same pattern, and so we can use the mouse as a way to develop these tools. I'd also like to say, you know, this is like kind of opening the candy store of tools here that this works subcortically as well. Here are examples of AAVs targeting the striatum, and in particular the indirect pathway in the striatum. We also have tools to target the direct pathway and the interneurons as well. And now we've begun to even push this down to this finest level of resolution in this molecular tree. So as I described to you earlier, this chandelier cell, this is a very distinctive type of cell, but really at this fine level of resolution. And we've now been able to generate the first tools to be able to target at this level of resolution as well. This is a beautiful image of the mouse cortex. There's a band of these chandelier cells up at the upper surface of the cortex. And all of these little vertical lines that you're seeing are these cartridges, these axonal cartridges that these cells form as they're connecting to the axon initial segments of the pyramidal neurons. Hot off the press, it turns out that one of these enhancers gives similar specificity in the monkey cortex. So now the cortex is expanded, these cells have expanded. You can see the wide breadth of these axonal cartridges across wide swaths of the cortex. And so it appears that this approach to use the molecular classification and the state of the chromosome and the regions of open chromatin as a direct path to identify tools to target very specific cell types, and that this is a very generalizable strategy that also works across species. Okay, I want to end with a final vignette, which is a bit more on the basic research science side, but it illustrates a number of points, and it really kind of brings together this whole toolkit that we've been working on. So the focus of this will be the deep projecting neurons in layer 5 of the cortex. In motor cortex, these have been traditionally referred to as pyramidal tract neurons. This is actually not a great descriptor of this type, because this is a type which is found across the cortex, and many of them don't contribute to the pyramidal tract. A better term is called extratelencephalic-projecting, or ET neurons, that project deep to the cortex, which differentiates them from the telencephalic projecting IT neurons, or intratelencephalic neurons. The transcriptome has shown a very robust molecular definition of these types within this taxonomy. And one of the things that we found as we first compared mouse and human was that the relative proportion of this type varied quite dramatically. It's about 20-fold less abundant in the human cortex. And it appears that the reason for this is explained by allometric scaling in the cortex, where the cortex itself has expanded much greater than its subcortical targets. And so in order to have a matching of the cortex and its targets, it seems that this particular type of cell has been sparsified as the cortex gets bigger. And this is sort of supported, at least conceptually, by the size of the different regions and the number of neurons in these different regions, that also are about a 20-fold difference of the disproportionate expansion of the cortex. in human compared to mouse. We've been able to develop genetic tools, these viral enhancers, to target this particular type of cell. On the left-hand side here is an example, a series of examples from mouse. The first were developed by my colleague Patch-seq, and then in my team, Jonathan Ting has developed another series of these to give very, very selective labeling of these types in the mouse. And these also work across species, and that you get a very dense band of cells in the mouse cortex, a significantly sparser band in the monkey, and very, very sparse in the human, which again supports this idea of the sparsification of these cells in the cortex moving from mouse to human. So these tools now allow us to target really for the first time some very famous types of cells using these types of techniques. So primary motor cortex, of course, contains the famous Betz cells. These are the upper motor neurons that project down to the spinal cord. And these are actually very difficult to study using patch clamp physiology because the motor cortex in monkey and human is very highly myelinated. And so it's very hard to come in and just use unlabeled techniques to target these cells. But now we can label them with a bright fluorescent protein, which allows us to target them and start to measure the selective properties of these cells. We were able to establish that the cells with Betz characteristics, such as this enormous morphology, they have this unusual downward tracing dendrite called a taproot dendrite, all mapped to this ET variety. So in the mouse, this ET are the corticospinal cells. Now we've been able to map these monkey cells. And in fact, we even got a very unusual surgical resection in the premotor area. And we're able to find several cells from this human premotor cortex that have very similar anatomy to the macaque and also map to these ET types. So the ET contains the Betz cells. And now we can compare properties across species. And what we sort of have typically finding on almost any kind of cell, it seems, is that there are a series of very conserved properties. So the differentiation in properties, for example, between the ET neurons projecting deep and the IT neurons projecting more locally are robust across species. And they can be clearly differentiated based on even a small set of properties such as the resonance frequency and the input resistance of these cells. And we can find ion channels that are potentially responsible for those conserved properties. At the same time, we also see very divergent properties of these cells as well. These cells can fire at extremely high frequencies and maintain that fast firing rate, almost looking like fast spiking interneurons. You can see this in particular on the right-hand side here for the macaque Betz cells. They also have this unusual property where they initially fire under constant current, and then they have a long pause, and then they start firing like crazy. And in fact, their firing rate accelerates, this property of firing rate or spike frequency acceleration. And this is not seen in the mouse ET neurons to anywhere near the same degree, or nor the IT neurons as well. And so these properties are actually very consistent with extracellular recordings from literature. In the cat, for example, the cats have a very robust population of the Betz cells, but also display this type of bursting high frequency behavior as well. But now we've been able to really show that these are the same types of cells or homologous types of cells that have some very specialized properties between both primates, human, and compared to the mouse. I want to highlight that this really opens up the first opportunities to begin to study a whole series of these types of cells that are both sort of textbook famous, but also disease relevant. So this ET type of cell not only includes the Betz cell in motor cortex, it includes the von Economou cells in the anterior cingulate and the insular cortex that are selectively vulnerable in frontotemporal dementia, as well as the famous Minard cells in the visual cortex. So So really, really opening up the first opportunities to do detailed studies of many of these types of cells and understand their distinctive features across species. So let me end now to step back out again. So this really is an accelerating enterprise here, and the Brain Initiative Cell Census Network has taken what I've just described in a single brain region, and the next phase of this is essentially whole brain. And the mouse efforts will be delivering at least the transcriptomic data on the entire brain in the next year. The human brain efforts that I'm leading are also creating sort of a draft version of about 100 regions. And this includes most brain regions across the human. This is a draft, I should say. We've also just processed our first spinal cord sample, which may be of interest to this audience as well. And the Brain Initiative has signaled that creating a similarly granular version of the human and probably non-human primate is a major priority for their next phase, as is trying to take this classification and turn it into a series of tools that can have impact both by opening up more model systems as well as potential disease applications to complement this using this enhancer type of strategy. And I think we can extrapolate at this point based on what we see in the cortex that this will be a very complex situation, but also a bounded one. There will be thousands of cell types that vary across the brain that make up all of these different brain regions. And I think the real promise here is that we now have a strategy that can really define this complexity. And I think in the next sort of 5 years or so, we will move beyond this discovery state of trying to find new cell types to really a deep understanding of that classification and using that then to begin to understand disease. And so I'll end with that point that really all of these tools that I've described to sort of build this basic reference are now available to try to study cellular phenotypes of disease, to screen for understanding if particular types of cells are vulnerable or resistant or proliferative, what molecular pathways are perturbed, and perhaps to lead to a different approach to thinking about therapeutics as not necessarily molecular targets as cellular targets. If we can target the right cell types and deliver the right payload, we could have potentially many types of treatments for different disorders. And just to give a little plug, we have now begun a big project on this premise to study Alzheimer's disease with our colleagues at the University of Washington and at Kaiser Permanente Washington Health Research Institute with their ACT program to try to bring to bear these single cell approaches to look for really consistent cellular phenotypes of disease and the molecular underpinnings of those. So with that, I'd like to end by saying that, that these are really big team efforts. As I've described, both at the Allen Institute and with these other bigger consortia, it takes very large groups working together to achieve these types of things. We have a really terrific team at the Allen Institute that contribute in various different ways. And I'd also like to end by thanking our late founder, Paul Allen. Thank you for your attention. I'd be happy to entertain any questions.
Hey, Ed, that was really terrific. Can you just talk about, I think one of the things that is really, I think what you guys have done, and I know your talk today was more scientific, but can you just comment a little bit, Ed, about how, you know, the Allen Institute in particular, kind of Paul's vision of how you guys share data and how you put everything online, because I think that's pretty revolutionary. You know, historically, you know, for all of us, whenever people have done research projects, they've had grants, you know, everything was kind of kept confidential and everyone waited till the, you know, ooh, don't tell anybody, we're going to publish this. And then it gets published in some obscure journal that nobody has a subscription to. And so can you just talk about that? Because I think that's really important. I think that's actually been you know, obviously there's a huge amount that Paul Allen has done in the scientific community, but just talk about how, I mean, I think he's such a visionary to kind of open up this old notion of how people do research.
Yeah, thanks for bringing that up, and maybe I should have made more of a point of that in saying that we do open science. You know, Paul really set the stage here for feeling that he wants these efforts to be really broadly catalytic for the community. And the real way to do that is by sharing this information openly and early. And this has always been a mantra for our own work, that we don't wait for publication. We do, of course, analyze our own data, but the idea is that we can get the best of both worlds. We can both make extremely useful community resources and also make discoveries on that ourselves. And you know, I will say there was, you know, a fair amount of consternation early about this idea that you know, we would get scooped a lot, and it has never come to pass, really. The types of things that we do are kind of broader in scope. And this actually, I think, ended up becoming one of the bigger legacies of Paul, that has really influenced the field very much to move in this direction, that everyone should be sharing data in that way. And the big consortium efforts that I'm talking about have adopted that type of mentality, as have the genome projects, I should say, that even preceded a lot of this. But so, for example, the cell census efforts that I was describing with NIH, everyone has to put their data into these data archives and make them available well before publication. So, so I think really this is the way to go for this field, that there's so much value for so many people from these data. And more there's value from integrating across these different data sets. It's possible to aggregate data across the different projects, that it's really essential for the field to be adopting this open access and early data sharing types of approaches. And you know, this is just a way of life for us now, but it's been really gratifying to see that the funding agencies have responded to that and started to make it really just the normal thing that is expected of publicly funded research.
Do you feel that your, you know, academic sort of affiliations with different institutions, do you think it's starting to affect the way they do research as well? Kind of the traditional, you know, for example, you know, if you look at historically, like, you know, larger academic institutions that were heavily dependent on NIH grants, do you think it's trickled down to those labs now?
Well, it certainly has, in the sense that to participate in these bigger consortium efforts, you have to agree to such a thing. It's kind of the state of things now. And so I think it differs for different types of data. For the omics data, this is just normal now, and it's you know, I think actually it's become a highly rewarding thing to do, because you get credit when people use your data. And so I think it's very much trickled down to that level and to those kinds of people using those kinds of methods. It has not trickled down, you know, super broadly to all other kinds of data that people generate. But I think that's in part because there aren't such easy places to put those kinds of data either. And so I think as NIH and others start to really better develop the data archives to house that data, I think that we're going to see that happening much more broadly.
Yeah, I mean, I think for us, I mean, I'm always going on the Allen website with the fellows, and it's incredible how much data you guys have. I mean, you guys must have an entire building just with servers.
It's a lot of data, it's true.
It's actually remarkable. And do you guys track? I mean, I'm sure that pharma, because this is what I think is revolutionary, is you guys were really the first, you know, high level group to put everything out on the internet. Do you guys track what, you know, like I'm sure pharma, you know, things like that. Do you guys track any of that data of people going to your website?
We do. Yeah, we of course we're able to track how many users and where their domains are, so we can see if it's coming from different institutions or different companies. And it's really had a pretty remarkably broad base of users across the world in different fields. And it's interesting actually that the publications are still really an important part of this. We put all this data out there, but it's not until the publication comes that there's sort of broad understanding of what's there. So it's not, it turns out to get people to use these data, you still have to go through the traditional mechanisms of illustrating what's there and what their utility is.
Yeah. Well, thanks again, Ed. We really appreciate it. And I know you guys have this thing called Open Science Week. But actually you guys are open science 24/7. So it's really, you guys have really been great partners, really, you know, it's inspirational work that you're doing. And I look forward to catching up with you in the next year. So you guys have done a lot since last time we talked. So.
Thank you very much. I really appreciated the opportunity.
Great. Thanks, Ed. Take care.
Okay. Yeah.
Okay. Bye.
Thanks for watching. Hit the subscribe button for more medical content.